Hi, everybody. Welcome to ANT 200, Introduction to Archaeology. Today, we're going to continue our conversation about Neanderthals by talking about the role of ancient DNA in the discovery of the human past. In 1984, geneticists recovered 229 base pairs of genetic code from a subspecies of zebra extinct since the late 1800s. The discovery proved that DNA could survive in dead things and spurred a new field of science called paleogenomics. Ancient DNA extracted from human bone and teeth can be used to determine the biological sex of an individual, familial relationships, and to identify the presence of disease pathogens and provide genetic ancestry. Today, technological advances in paleogenetic research allows archaeologists to read the genomes of ancient humans and other organisms, transforming our view of history and evolution. Most ancient DNA comes from samples younger than 50,000 years old and from very cold climates. When organisms die, their DNA slowly begins to decompose. How long this process takes depends on factors like temperature, burial conditions, and the number of microbes decomposing in the body. The general estimate is that optimal, very, very cold conditions allow DNA to survive about 1 million years. To date, the oldest complete genome belongs to a horse unearthed from frozen ground in the Yukon here in Canada and dates between 560 and 780,000 years old. The oldest DNA from our human ancestor is a fragment of genetic code from a 430,000-year-old Neanderthal found in 2016 in Spain's Cima de los Huesos cave. Geneticists are starting to have success with aDNA preserved in warmer places as well, which is particularly important for understanding human evolution in Africa. In this image, you're looking at a map that shows the location of human remains that have yielded whole genomes that can be used to determine ancient DNA samples. In the early days of aDNA testing, geneticists could only read very short stretches of ancient genomes. As a result, studies really largely focused on tracing either male Y chromosomes or mtDNA, mitochondrial DNA, which is produced by the maternal line. These narrow portions of DNA don't reflect an individual's complete ancestry, which can only be determined by gathering DNA from across the full genome. Obtaining DNA from all 23 chromosomes is fairly straightforward for modern humans, but poses a significant challenge to extract and sequence an ancient DNA. This is because DNA degrades into fragments that become undetectable while also undergoing chemical reactions that change its code over time. Moreover, aDNA is typically found in trace amounts within archaeological samples and is often masked by genetic material from the variety of microbes teeming in many research environments. One of the major problems that prevents the widespread sequencing of hominin ancient DNA for several years was the issue of contamination. Genetic material extracted and sequenced from living individuals will consist largely of DNA fragments from that individual. In contrast, ancient DNA is so scarce and fragmented, most of the genetic material extracted from fossils tends to be environmental microbes or from humans who have handled the fossils themselves. This human DNA is particularly troublesome since some present-day DNA is similar in sequence to ancient DNA from fossils. Geneticists have developed two main ways to address this problem of contamination. Now it's standard practice to extract ancient DNA under strict clean room conditions with a filtered air system and UV radiation to minimize the proportion of outside DNA in a sample. Geneticists have also begun to sequence several bioinformatic markers that can be used to remove contaminants or estimate the proportion of those contaminants present in a DNA sample, and then to exclude them from analyses. So how do geneticists even analyze aDNA? Well, the first step in the processing of aDNA is extraction. In a sterile lab, bones and other surviving tissues excavated from archaeological sites are cleaned, crushed into a powder, and then dissolved with chemicals that isolate short DNA strands. Researchers add tag molecules to this extracted sample that work kind of like a barcode or an SQR code. These tags identify any additional molecules accidentally added during the initial processing of the sample, as well as identifying any subsequent sequencing steps from being confused with the molecules that are actually within the DNA sample itself. To read the DNA, computers analyze millions of DNA copies simultaneously. These copies are made by geneticists through a process called polymerase chain reaction, or PCR for short. The basic idea in PCR is that 99.9% of our DNA is the same. But we also have unique variations that can be used to identify an individual. PCR makes billions of copies of the DNA sequence that you want to look at, making it possible to find variations. Without making lots of copies, you can't really detect the sequence that you're actually interested in. It would be like trying to hear a single instrument in a large orchestra. So what exactly does PCR do? Well, polymerase is a protein that makes copies of DNA in cells, and they use this polymerase to copy DNA to make more different copies that can be analyzed. The chain reaction of polymerase describes how each time polymerase, that protein, makes a copy, the number of copies doubles. Repeating this process makes 2 strands into 4, 4 strands into 8, etc., etc. After 35 rounds of making copies, PCR produces 34 billion pieces of targeted DNA. In the final round of PCR copying DNA, geneticists begin to sequence that DNA. Sequencing means determining the order of the 4 chemical building blocks of DNA called bases. These are signified by the letters A, T, C, and G. In the DNA double helix, these 4 chemical bases always bond with the same partner to form what's called base pairs. A matches with T and C with G. The sequence tells us the kind of genetic information that is carried in a particular DNA segment. The human genome itself contains 3 billion base pairs that spell out the exact instructions for your particular genetic makeup. Computers then read these base code pairs, analyzing all the identical strands with matching tags, at the same time weeding out any errors. Researchers use a number of clues to discard contaminants in these ancient DNA codes. For example, aDNA strands are usually shorter than 100 letters and have predictable degradation patterns. The short strands that have been read need to be arranged into their proper position across a full genome. Software lines them up based on overlapping stretches of code and comparisons with previously sequenced reference genomes. In this sense, our genomic information is only as good as the sample size that we have. For anthropologists, ancient human DNA provides insights that we couldn't learn from fossils or artifacts alone. It's already helped provide answers to some major debates, including whether modern humans interacted with Neanderthals. Despite overlapping ranges of Neanderthals and modern humans in Europe and West Asia for at least 100,000 years, there was no widely accepted archaeological evidence that they had interacted or interbred. A 1997 study found that mitochondrial DNA, that's the DNA that we get from our mothers, remember, that these mtDNA for Neanderthal fossils lay outside the maternal DNA sequence for modern humans. Well, this study showed that there was no admixture between Neanderthals and Homo sapien groups. It didn't prove conclusively against it. Nuclear ancient DNA from Neanderthals resolved this problem, and its analysis showed that actual admixture was different from what had been expected previously in discussions of Neanderthal and Homo sapien interaction. A study of 3 individuals from the Vindija cave in Croatia provided the first comprehensive evidence of Neanderthal genes. These results were compared to genomes from archaeological remains associated with individuals from France, China, and Papua New Guinea, as well as from 2 African populations. The data showed that the Neanderthal genome was more similar to non-African populations than to those from Africa. This difference was because Neanderthals and their ancestors and the ancestors of present-day non-African populations had interbred in the Middle East, where the ranges were known to have overlapped. Further comparison of the Vindija Neanderthal genome with a larger number of present-day human genomes has revealed the surprising fact that the genetic similarity of East Asians to Neanderthals is greater than the similarity of Europeans to Neanderthals. These ancient genomes show that our ancestors not only met, but mated with Neanderthals between roughly 40,000 years ago and 100,000 years ago. This intermixing was so common, in fact, that Neanderthal inherited genetic material is found between 1 and 2% of all non-African populations. Ancient DNA research has complicated this history of species interbreeding even further. In 2010, mitochondrial DNA was extracted from a pinky bone found in the Denisova cave in Siberia. When this DNA was sequenced, it was discovered that this pinky bone represented the DNA of a sister group to Neanderthals that had diverged from them after the ancestors of modern humans had already diverged from Neanderthals about 500,000 years ago. Scientists refer to this group of human ancestors as Denisovans, after the cave in which the remains were discovered. Denisovans were the first archaic hominin group that was characterized completely on the basis of genomics, since the fossil remains were actually too scarce to provide any morphological or physical information. Genetic research has shown that Denisovans had a different history of admixture with modern humans than Neanderthals. Denisovan ancestry is found in Melanesia and in native Australians, and to a lesser degree in peoples from East Asia. The pattern of admixture suggests that they once had a broader geographical range than Neanderthals, who primarily inhabited Europe and Western Asia. Genetic testing of a tooth found in the same cave showed that Denisovans were also more genetically diverse than Neanderthals. Comparisons of Denisovan and Neanderthal genomes led to the conclusion that Denisovans had mixed with Homo erectus and other archaic hominin groups, which diverged from Homo sapiens about 1 million years ago. The first complete genomic sequence for Native Americans was published in 2014 by Rasmussen after testing human remains of a young child identified in Montana by a girl named Sarah Anzik in 1968. The individual referred to as the Anzik 1 was associated with a large cache of ochre dust stone and tools, particularly Clovis points, that date to about 12,600 years old. These remains were directly ancestral to present-day Native American populations. In 2015, Rasmussen's team conducted genetic tests on these human remains, known as the Kennewick Man. As we'll get into, these remains have engendered historical and legal questions about the genetic origins of indigenous people. ADNA has helped archaeologists answer questions about the timing of human migration into North and South America. In more than a dozen studies, geneticists found that Native people in the Americas stem from 4 major maternal haplogroups. These are kind of our basic human lineages. Only the modern inhabitants of southern Siberia match this genetic profile. A large-scale genomic study of 31 present-day humans and 23 ancient human genomes from 200 to 6,000 years ago has demonstrated that all Native peoples in the Americas separated from their ancestors about 20 to 23,000 years ago. Northern Native American peoples diverged from their Southern and Central American counterparts roughly 13,000 years ago. This research also showed that there was no single wave of immigration of ancestral Native Americans from Siberia. Instead, Native Americans received significant genetic gene flow from East Asians and Austro-Melanesians, especially among Southern Americans. So let's jump into a little bit more about the Kennewick Man and the role of ancient DNA in discovering the Kennewick Man's ancestral origins. In July of 1996, 2 college students stumbled on human remains that had washed up along the Columbia River near Kennewick, Washington. The skeleton came to be known as Kennewick Man and was 90% complete, representing a male standing about 5'8" tall and weighing roughly 170 pounds. Subsequent forensic testing revealed that these remains were more than 9,000 years old. A storm of controversy erupted when the Army Corps of Engineers, which managed the land where the remains had been found, claimed authority over the Kennewick Man remains and demanded that all scientific study cease. At the same time, a coalition of Columbia River Basin indigenous tribes claimed the skeleton under the Native American Graves Protection and Repatriation Act. These communities demanded that the remains be handed over for reburial, and the Army Corps of Engineers actually agreed. These indigenous tribes had good reason to be sensitive and angry about the collection of their human remains by archaeologists and anthropologists. During the 19th century, anthropologists and collectors looted indigenous graves, excavated their remains, and even decapitated indigenous peoples killed in battles, and shipped their remains to Washington, D.C. for further study. In response to the Columbia River Basin Coalition's repatriation request, 8 anthropologists filed a lawsuit to halt the repatriation process. The scientists argued that these human remains had to be studied in order to determine if they showed any different affiliation to modern indigenous communities. Before repatriation can occur under the Native American Graves Protection and Repatriation Act, indigenous communities must demonstrate a cultural affiliation between those human remains and their own groups. Because of the age of the Kennewick Man remains, the scientists challenged the cultural and historical evidence for connection. The court confirmed their argument, ruling in 2002 that these remains were not related to any living indigenous community, and therefore that the Native American Graves Protection Act didn't apply to the Kennewick remains. The court, this is a ruling by Judge John Jokers, who ruled against the Kennewick Man repatriation in 2002. Almost 10 years after the skeleton remains in Kennewick, Washington were found, scientists were given 16 days to examine the remains. In those 16 days, a team of 22 scientists scrutinized 300 bones and fragments, analyzing the remains using 3D images to create a portrait of Kennewick Man displayed here. The scientists argued that this portrait did not look like modern Native Americans because the remains had a much narrower skull and smaller face. This led to a series of claims by white nationalist groups that the original inhabitants of the Americas were actually of European ancestry rather than indigenous. Subsequent studies suggested, however, that the closest living relatives of Kennewick Man seem to be Maori people from New Zealand, as well as people from Japan, indigenous people from Japan called Ainu. Hoping to find a more definitive type of evidence for Kennewick Man's identity, scientists also called for genetic analysis. Recent studies have thrown Kennewick Man back into the spotlight. In 2015, DNA analysis of the Kennewick remains revealed that his genomes were more loosely related to modern Native Americans than any other humans in existence. This overturned the previous court ruling that the remains were not culturally affiliated with Native peoples and gave the coalition of Native tribes from the Columbia River Basin the right to rebury their remains. The Kennewick Man controversy brings up a central worry for indigenous peoples around cultural affiliation. Basically, should genetic definitions of relatedness prevail over indigenous definitions of knowledge claims? In your analytical assignment for this week, you'll tackle this question around the Kennewick Man controversy.